Hi everyone, my name is Aiman, and in my project I'm visualizing the recurrent structural variants that are affecting tumor genomes using GROM and Variant Navigator. So for some background knowledge, structural variants are basically large genomic alterations that are often responsible for tumor development. Most studies will focus on single nucleotide variants, while we are actually focusing on structural variants. So to do that, we use two main computational tools, and our whole project is fully computational. The first tool is the Genome Rearrangement OmniMapper. This was developed in the lab and is used to identify the variants. The second tool is the Variant Navigator. This is used to visualize the variants. So the question that we're asking here is, do recurring structural variants in genomes of multiple myeloma patients affect tumor genesis? We hypothesize that these structural variants do play a part in the development of tumors as they overlap certain genes that have functions related to cancer symptoms. So to test our hypothesis, we use whole genome sequencing data of multiple myeloma patients that was produced by next generation sequencing technologies. We carried out multiple steps that include sequencing the reads with Illumina. Reads are basically small DNA fragments, and then aligning them with the reference sequence, as you can see in figure 5. Next, we analyze with GROM and filter them using Java code, and then visualize them using the Rain Navigator. When we were visualizing, we were basically comparing tumor and normal samples, and we were looking for differences in the variant size and the genotype meaning homozygous or heterozygous, and also looking for common overlapping genes. So in figure 6a, you can see there's a difference in the genotype. So the bottom one is basically homozygous and the top one is the heterozygous one. In figure 6b, we can see there's a difference in the variant size. So these are deletions. The top deletion is much shorter than the bottom deletion. Another thing we can see in figure 6b is the pink and yellow lines, which are basically genes that are overlapping the deletion. So after doing that, we record our results on Excel and we form merge intervals by combining multiple structural variants that have similar coordinates. So merge interval goes from one coordinate to another coordinate on a chromosome, and if we look at multiple patients that have similar variants that are occurring at similar coordinates, right? So we would combine them in one single merge interval and visualize them as it is seen in figure number 7. So the orange ones are the tumor samples and the blue ones are the normal samples, and we group them all up of multiple samples. And we can see that the tumors are actually grouping up and breaking at one single coordinate. So this would mean that basically there's something similar happening in a bunch of multiple myeloma patients and it's at a similar coordinate. So this could mean that this is basically causing tumor development. So we visualized 597 deletions of 18 patients, and from all of those, we found multiple merge intervals, but the most interesting ones are the ones that I've listed here in this table. There are 6 of them. As you can see, there are colors pink, yellow, and gray. So the pink means homozygous deletions. I've grouped them up into their genotypes. So the pink is the homozygous, and the yellow is the heterozygous, and the gray or the 0 is just no deletion. So for example, in the first merge interval that I've listed here, the tumor, for example, we see the sample 4091, and for its tumor it had no deletion, and for its normal it had a homozygous deletion. You can also see genes that are overlapping or nearby these merge intervals. So the first one is nearby the gene LRP5, the second one is near it's overlapping IGFN1. The third one is overlapping and also nearby the GLB1L2 gene. Then at the bottom table number 2, we see functions of those genes, and we can see that those functions are related to cancer and cancer development. So these are the genes that we discovered. And I would like to thank the Summer Undergraduate Research Grant, the Center for Computational and Integrative Biology, the Rutgers Biology Department, and the Grigoriev Lab for giving me the resources to take forward this project and work on it. For future directions, I'll be analyzing different variant types like inversions and duplications. So these ones were all deletions. And I'll be exploring more data to collect stronger data for already located genes and discover new overlapping genes as well.